All right. Hi, everyone. Welcome to the first ZEISS Micro Minute. My name's Nathaniel Cohen, and I'm an applications engineer here at ZEISS Innovation California, working at the ZEISS Microscopy Center here in Dublin, California. So today we'll be looking at a little adventure into what is correlative microscopy, where we can take two different modalities microscope, at least two different ones, and we can correlate them together. So our first subject, our only subject today, at least, diving in an eggshell, standard everyday eggshell. In fact, this is eggshell that was from my eggs in the morning, and I took the egg in the lab and on a couple of our. So before we get into too much detail, I want to show you a video I produced. This came out of our ZEN core software. That's what we'll be using and at later today, how we did our correlation. So without further ado, let me play this quick little video. I'll narrate over through. So here we are. I'll pause right now before it dives in. The lowest magnification picture, if you want to call it that, didn't come from a microscope. Actually, a photo from my cell phone that I imported into the software. And now we'll be diving in and going first an image from an optical microscope, our Axio Zoom, and then higher in magnification and look at very tiny structures that we the egg. So I'm going to go ahead and press play now. Our zoom in. There's kind of the field of view from the optical microscope. Now we can see the region of interest that I zoomed into, this core that was on the surface, increasing, of course, some cracks. Then what becomes apparent is the surface. It seems to be making lots of little spheres. It was honestly quite surprising to me. And here we can see the scale of these spheres. Scale bar on the bottom, that's 1 micron. So these spheres, nanometer scale, 1/1,000,000th of a meter. Quite tough. Here's another region that seemed kind of interesting where it seemed like that outer layer torn away. And we can actually see one level deeper into the eggshell where the spheres are just starting to form. Now that's a top-down view. Let's zoom out. And what does it look like then from the side? So if we then zoom in to a portion that we mounted vertically, we get a cross-sectional. We actually could see there's a membrane layer. Look at that. The structure is totally different, looking fibrous. Zoom out again. Now we're going to go into the bulk of the material. As we increase in magnification, actually start to see pores here in the cell. Once again, these are down at the nanometer scale, really only able to be with electron. And then we'll zoom back out. There we are. There's the video. So here I am. Hello. This now I'm in front of is ZEN Connect. So this is the magic of how we made that video and how we can do correlative microscopy. So in this environment, we're seeing multiple images, all the data that we've collected. If you've used any other kind of audio, or rather video editing or Photoshop-like program where you could bring images, here's our kind of layers, what is displayed what. And we have data from two distinct sources. We have some imported images. We have images that come from our different microscopes and different sessions. So let's push in a little bit. I could hide this. And so here we see the overall eggshell. Now, to give you a little bit into the movie magic of what you saw, this eggshell that I took a picture of, I did that before preparing the sample. I had the eggshell in my hand, took a photo of it with my smartphone, and I noticed that this part was sticking out, was kind of obvious. So if I bring back my layers, fade it in and out, so we can see underneath, that part was attached to the egg. So kind of the movie magic here is that I took that piece of eggshell, and that's what I then prepared on microscope stub, and then brought to our Axio Zoom, an optical zoom microscope where we can use light to inspect the surface, and that's where we see here. And then I took a higher magnification. And if you had, let's say, imagine you had a really, really good magnifying glass, or in our case, a really good optical microscope. This is what your eye, this is just with natural light, where we can see kind of the nooks and crannies, some cracks, some little spots here on the egg. And this was the region that I then did this electron microscopy on. So just a quick note, before we take a deeper look at what we saw from the electron, I want to point out that there's a history here, right? This isn't just you're looking at eggs for fun. I did a quick little literature review on some egg structures, and I found, even going back, a paper from Carl Hirsch and Packard from 𝗠cie𝗿𝗄, or rather, not sorry, Colorado State University. This is back in 1986. And here they were looking at some fossilized eggs, talk about the structure of eggs. Then if we scroll down a little more, we see that they were using electron microscopy here to help understand what the structures of were. And these are actually fossilized eggs. You can appreciate some of the electron micrographs they take here. Now, a little bit closer into the present, great overview of the structures. I'll come back to that figure in a second, but this is from a review paper from 2012 from Maxwell-Hinkie et al. Coming out of Ottawa. They've made this excellent paper going over the structure, composition, and mineralization of the eggshell. So if I scroll down, it talks about how eggs are made. And here we see some of their electron micrograph. And some of these details, as we saw those pores as well, this is of interest and study in. And so just so we can put some right terms on this, so if there are any egg experts out there, they don't get too mad at me for not using the right terms. So hopefully I'll use the right terms as we go through. So that top surface of the egg we are looking at, that will be called the cuticle. And it is noted here in the paper that it is spherical small forms. And one of the structures I zoomed in for, and those pores actually can go down through the bulk of the egg, on that layer they call the palisades. And that's where we'll see those tiny pores reach well. And they end in these mammillary cones and then go to their external shell membrane. So now let's go back to my data and see if we can plot these same structures. Going back to ZEN Core, here we can see what was in optical microscopy, just looks like little divots. And in fact, I believe this is one of the larger pores that we see. So bringing this into the electron microscope, I should note that no other preparation was done. This was brought into our Sigma field emission SEM, and without doing any metal coating or anything like that, this put in as is. And looking a little bit higher in magnification, we start seeing some of the cracks we saw. You get even a full view here. And then coming in a little bit more, here we see these spherical structures that we observed really to a small scale. Now, reading through some of the papers, so in the cuticle here, proper term, at least according to that paper, I couldn't find a more recent one. So if there is any egg experts out there, I want to say eggsperts, I did, that from the paper I was reading, it's not clear exactly why they have this structure. One idea was that it somehow made it actually more difficult for pathogens to hold onto. Obviously, I think it gives it a little bit of resistance as well, maybe so they're not so smooth. But what I want to look at, if we're looking at that structure, we need to look at the cross-section. So if you're familiar with using a mapping program like Google Maps or something, that navigating around your data space here is just the same, right? That I'm just using the scroll wheel, panning across, and all the data is in context with it. I'll give you a little peek behind the scenes of how the software is working. Our microscopes, when they take images, it knows the size of the image and it knows where the picture was taken. So if you're doing this all-in-one imaging session, you just import your data. These images are automatically stacked and positioned. So for example, all of these data that I collected in the microscope was done in one session. And when I import it, these groups of data are all registered itself. I'll show you in a little bit how we can get it registered in a larger image. But let's take a look at this cross-section. And here we actually do see structures that paper is talking about. Cuticle on the outside, and then this palisade region. And then, as it mentions, mammillary cone structures at the bottom, you can actually clear those structures as well. And then that inner membrane out of softer kind of proteinaceous materials. And if you've looked at paper at high magnification, I have in the this does look very similar to how paper or other membranous structures look like. Nice structure to that. And if we come over to this palisade region, then this is where what really shocked me, that we see these pores and all the way down to this nanoscale. So if we look on the bottom, we have this universal scale bar that follows us as we're that has our scale bar, gives us the idea of the size of the field. So right now, this entire field of 16 microns across, or a pixel on my screen is now 10 nanometers. We can also appreciate this calcite structure to the egg, and where we really can see the cleavage of how that broke of this eggshell. So all in all, this eggshell, while incredibly resilient, highly structured, right, about 3 microns in thickness here. That's about the same as a thick human. Let me zoom out, and now let me talk about how we made a little bit about the correlative microscopy. So I already mentioned, right, this was an iPhone. That doesn't have any pixel scaling, but what did was this initial image I took from the microscope. Now, with that image, I would position this iPhone image into where we see this. So to give you an idea of what that looks like, if I bring back my panel and I select that iPhone image, we have this tool to allow us to align. So it's already aligned. See, I can move it around and so how this was actually done, if I take this image and make it just a little bit translucent, select my iPhone image, press align. This picture was much smaller. I needed to reposition it. So we have different tools that allow us to. And then this is really the process. See underneath, get it to fit, then you would be able to align those data. So that's how I was able to get that iPhone image scaled, because I was something that had the proper scaling. Now, just like I mentioned on the electron microscope, on the optical microscope, that once I zoom in and take another image at higher magnification, those are registered with each other. But now here we see we have a lot more resolution from that optical image. So that's the optical to, let's say, other source, or any JPEG TIFF from any source, and scale it to what we see here. But now how do I do the correlation between the optical microscope and the electron microscope? We have several different methods to do it, but the easiest is really just taking the sample and putting it right into the SEM. And as long as your sample has some obvious features, I can find those same features as I navigate around on my SEM, take images of those, then as I import the data, I can match them. And in fact, I have this software running on my SEM that allows me to do this correlation while I'm sitting live. And so the points I use for correlation, we see these SEM images from the tip, then this other part over here on an edge, and then this final edge. Features are obvious between the two. Once I have those 3 points, I can align the optical data to where I'm at on my SEM, and then allowing me to use that optical information to navigate to this, start collecting. And then I proceed as I would doing standard SEM operation where I'm finding, navigating. So one question I had, actually, I hide some of these layers, hide this one. One of the things I spotted from the optical data was this little red spot. I was wondering what it was. So I already had looked at kind of what pore space was, and then I wanted to say, okay, here. And when I took a picture of this region of interest with the SEM, now I can see overview, and then I saw where that red dot was. So if I'll fade this in and out again, you can see that red dot was positioned there. Zoom in, then this is where I took another image. And it turns out that it looks like it's some other random crystalline piece. It could be a piece of the eggshell. I have a good answer for you, but it is whatever that little red spot was. Maybe later video we could follow up, or if there's questions about what this, dive back in and get that for you. Okay. Let's take another look at this side view here, because I do want to show I am hiding the best looking part of this, of where we had axio zoom, do a tight focus, a little bit of a depth of focus imaging here. So for those who are familiar with optical microscopy, when you're at high magnification, your depth of focus can be very thin. As you adjust focus, you can see parts, but you all in focus time. But if you do a extended depth of focus or a Z-stack, we're able to take a series of images, different focal heights, and then bring them all together. And that's what we see. We get the individual, this piece of data here as well, where we had a really quite nice optical bit of this eggshell. And we could actually see some of the same structures that we saw under the electron microscope match those structures here. So just remind ourselves of these terms again, that there's that cuticle, that crystal layer, the palisades, and the membrane. I think I can see those quite nicely here in optical data. And what's neat is that very thin membrane on the inside of eggshells, it can be kind of translucent when we look at it here with optical light. Now we come back to our ZEN Connect, bring in this EM data. This is where why correlation matters, that I can say with confidence on the same area looking at structures. And then we brought in this one here. We can then navigate through these different. So that's how I built this data set between these two different kind of the last thing I want to show you is also within our software, that video I started off with, we can also produce those videos here within our software. So once you build your ZEN Connect environment, you can come over here to your video maker. We could select what resolution of video we want to produce, say a 720 video. And let's say I want my video to start off here. And all I could do is say keyframe. We've turned off some of those data, but here, keyframe, and then there's this little red speck. Now, remember, in electron microscopy, we don't have color. We're only electrons, so it's all black and white. Then we could zoom back out to show. Now, this is going to be a little shorter of a video than the one I produced, but I just wanted to show you how easy it is. Because if you need to communicate your data to someone else, this is a great way to do it. And then I could say return to the first on end. Say preview. And then here's the video. So just letting this play through. And if you like what you see, you can then export it to an MP4. You have the ability to adjust kind of time that it stays in any one place, how long, different regions of interest. And here we are going into the cross-sectional view, zooming in, seeing that palisade. So that's what I wanted to show you guys. Hope you enjoyed going into eggshell understanding of how we do correlative microscopy and how we put it all together here within ZEN Connect, part of our ZEN Core program. If you're listening and you have any questions, there is a chat area that you can type into and put your questions into chat. We have some time to answer some questions. Love to hear from you. If not, send us a message later. we'll be able to address those. So if you're out there watching, thank you for your attention. And yeah, have a great day. I'll hang around just for a little bit, maybe zoom around this data, see if anyone has some questions. I have a little time. All right, there's that there. Okay, well, yeah, so thanks so much for your attention, and thanks for joining our very first Micro Minutes here. Hope to see all of you back here again. Tell your friends, like and subscribe, all the socials. Thank you. Have a great Okay.
Or to leave it up.